Hello, everybody, and good evening. My name is G Cajulao, and I'm a master's student at San Francisco State University in the lab of Dr. Erica Sanchez. We study Kaposi sarcoma herpesvirus, which causes the cancer known as Kaposi sarcoma. And in particular, we're interested in the role of a single receptor encoded by the virus. Now, what is Kaposi sarcoma, or KS for short? Well, KS is a cancer of the endothelial cells. When the cells that line your blood vessel start to divide uncontrollably, they show up as red bumps or lesions on the surface of your skin, as you can see in the image on the right. KS was very prevalent and important during the 1980s and 90s during the AIDS epidemic because it baffled researchers that a cancer normally thought to be endemic to the Mediterranean and North Africa was showing up in places around the world. Upon sequencing the tumors from biopsies of these individuals with AIDS-associated Kaposi sarcoma, they discovered a new herpesvirus. This herpesvirus, termed Kaposi sarcoma-associated herpesvirus, or KSHV for short, turned out to have two life cycles. It has a lytic cycle in which new viruses are actively being produced by our cells, and then it has a latent cycle where the virus's genome just hangs out tethered to our DNA. I'm going to be studying a gene encoded by the virus's lytic cycle. Now, throughout history of coevolution, KSHV actually stole a gene from humans that encoded what's called a G Protein Coupled Receptor, or a GPCR, which I'll explain in a bit. Now, throughout the course of coevolution, KSHV has modified this receptor. So this viral GPCR, what's unique about this is that it does not need a stimulator or an agonist to pass some signal down to the cell. Now, what is the signaling that I'm talking about? GPCRs, even in humans, are a large and diverse class of membrane receptors that are responsible for various kinds of functions within the cell. In fact, their functions are so diverse that they are the largest target for pharmaceutical drugs, which comes into play later on. Now, vGPCR does not need an agonist or a signal to tell it to pass some type of signal down to the rest of the cell. And it is also known for human GPCRs that this signaling triggers the GPCR to actually be physically pulled down and brought within the cell, where it moves and localizes to various locations within the cell. And this trafficking and localization is very important for how it signals. It's unknown, however, if KSHV's vGPCR exhibits the same phenomenon. We don't know how it moves in a cell, and we also don't know how important this movement is for the signaling of the cell. So far, to investigate this, I've generated cells that stably express the vGPCR for preliminary experiments. Now, since KSHV infects endothelial cells, I will move to endothelial cells in the future to confirm findings. Now, my immediate first step is to ask, where is the receptor in the cell after signaling? Using fluorescence microscopy and antibody labeling, as well as Western blots, I will determine the proteins that vGPCR interacts with, as well as the localization of vGPCR at various time points. Also, I'm going to be using luciferase reporter assays to determine how important each location is in the signaling of vGPCR. I do predict that trafficking happens, which is similar to human GPCRs, because again, this is just a modified version of a human receptor. I'm also predicting that this trafficking modifies how vGPCR signals and modifies the cell's physiology. This research is important because if we outline in detail the nuances of vGPCR signaling, it should inform future drug development and intervention. GPCRs, as you might have remembered, are the most common target for pharmacological drugs. So with that, I just want to say thank you to the ARCS Foundation and the donors for this opportunity.